Okay, so we're back with the second installment to the chapter on cell membrane structure and function, and we're picking back up with the membrane proteins. Remember we said we had the phospholipid bilayer and we had all of these proteins that were going to play an integral part in transport across that membrane and some of the signaling functions. So just review, here are your 6 major functions for membrane proteins. Remember, we're talking about proteins in the membrane, not necessarily proteins in general, but just those that are associated with the cell membrane. Don't forget your terms like selective permeability, amphipathic, glycoprotein, glycolipid, those guys. So glycoproteins and glycolipids, and I'm going to review those right here. So when I have a carbohydrate that is covalently bound to a lipid, I call it a glycolipid. Glyco for carbohydrate, lipid for lipid, obviously. They're relatively common when we talk about plasma membranes. More common, however, are glycoproteins, and those are going to be carbohydrates covalently bound to the proteins that are embedded in those plasma membranes. Now, the question is, why? Why do I have these? Why do I need these? I've got a protein, I've got phospholipids, why do I need to stick a carb off the edge of them? Well, the carbohydrates on the external side of the plasma membrane act as signals. They are signals to molecules, they're signals to other cells. They serve about 4 out of those 6 functions that we've already talked about. They don't really play a role in intercellular junction, and they don't play a significant role in attachment to the extracellular matrix, but they do play a role in just about everything else. So the other thing to remember here is that they'll vary. So the glycoproteins, the glycolipids, they'll vary among a species. They'll vary from individual to individual within a species, and they'll even vary within the cell types of different individuals. So liver cells are going to have different glycoproteins and glycolipids than liver cells, cardiac cells, skin cells. I think I said liver cells twice, sorry about that. But they're going to vary from cell to cell. So go back and remember selective permeability, because that's what we're talking about here. This is all about regulating the traffic into and out of a cell. Which molecules go in, which molecules go out, when do they do it, how do they do it? So remember your definition of selective permeability, and remember that the reason we have this is we have to maintain different concentrations on different sides of this membrane. That's going to be key when we start talking about diffusion and when we start talking about transport in general. Okay. Remember your terms for hydrophobic and hydrophilic. That's super important here, because hydrophobic molecules, those non-polar molecules like the hydrocarbons, are going to easily dissolve through that lipid bilayer. They're going to be able to pass without any significant restrictions placed upon them. That becomes key in a lot of maintenance situations for our cells, but it's also a toxicity issue. So some of those pesticides, those chemicals in our environment that affect our wildlife, that affect people, the organophosphates, for example, any of those hydrocarbons are going to move in and out of cells very, very easily without a whole lot of resistance, and that just increases their toxicity. Polar molecules, things that are polar, remember your definition of a polar covalent bond. So polar molecules like sugar don't cross that membrane easily. And I think that's actually rather crazy ironic, because those cells need that glucose to drive cellular respiration in the mitochondria, and yet we have to work to get that sugar into the cell. So kind of backwards. Try and keep that in mind. Transport proteins. So something like that sugar, something like those polar molecules that can't pass through easily, how am I going to move them across? And one of the methods for moving them into the cell are transport proteins. So transport proteins are designed to move hydrophilic molecule substances across that membrane. Sometimes they're called channel proteins. We'll talk about a very specific channel protein for water. Sometimes they're carrier proteins. So your 2 big classes of transport proteins are going to be channel proteins and carrier proteins. Channel proteins are just like they sound. They are a protein with a hollowed out center. Okay, they have a channel or a hallway or a tube, and that tube is hydrophilic in nature. It's lined with hydrophilic molecules, and what it does is it allows hydrophilic molecules or ions to pass through that tunnel. A very specific one that I said we'd mention is called an aquaporin. Aquaporins are specialized channel proteins that only allow water to move into or out of the cell. The other big group that we mentioned were carrier proteins. Carrier proteins bind to a molecule and then they change shape to shuttle them across the membrane. So it's like having a trap door. When the right key or the right piece fits into the trap door, it springs and opens to the opposite side of the membrane. Once the molecule releases, it snaps back and you're ready to load again. The other key part of this is that transport proteins are always specific to the substance they move. So aquaporins aren't going to move things other than water. They're only going to move water. A channel protein for sodium is only going to move sodium. It's not going to move potassium, even though I could have a sodium ion and a potassium ion both with a single positive charge. It's going to be very specific for the molecule that it's supposed to move. All right, there's one of your whiteboards. You're going to pause video, write down your 6 major functions to membrane proteins. I'm going to keep going. I'll count to 5, and then I'll move on to the next slide. There are your answers, straight off of the other slide. They're just listed here, but what you should be able to do at this point is you should be able to picture this in your mind. You should at minimum be able to think about specific examples for transport proteins. You should be able to put together the glycolipids and the glycoproteins for cell-to-cell recognition and signal transduction, and you should have a vague idea, if not specific, but a vague idea about enzymatic activity just from our conversations about enzymes in the macromolecules chapter. All right, diffusion. Diffusion is a huge topic. Big, 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 big. You want to have diffusion down solid. You want to understand what you're talking about, why you're talking about it, what's going on. This is something you need to understand for this course and as you move forward. So diffusion, by definition, is this tendency of molecules to spread out evenly within an available space. Okay? They don't cluster in one corner or the other. They spread out relatively equally across whatever space is available to them. Now, this doesn't happen because these molecules are sentient and they recognize each other and they recognize each other's personal space and, oh my gosh, you're too close to me and I've got to move. No, that isn't what I'm talking about. What I'm talking about is random movement. And we talked about that when we spoke of kinetic energy and particles moving and they're bumping off one another and they're bouncing around. And that's how diffusion is going to work. So even though these molecules are randomly hitting each other and bouncing, we're going to see a net movement in one direction or another. We're going to see a net movement from where they're more closely packed together, or that's often called a higher concentration, to an area where there are fewer of them in a close confined space, a lower concentration, until what we're going to get is we're going to get equilibrium. Equilibrium is when I have an equal number of particles basically spread evenly throughout. Now, just because we reach equilibrium does not mean that the motion stops. So in the upper section of these, we have diffusion of one solute. You've got that dashed line down the middle is a membrane, and it's kind of dividing that space in half. And in the first one, you have all of the solute, all the little yellow dots on one side of that membrane. And they're really close together. And because they're really close together, they bump into each other really frequently. And just the random motion of them is going to push some of them to the other side of the membrane. Now, as I'm in the middle image, now as they move to the other side of the membrane, I have fewer over on the left. They're still bumping and running into each other, but their frequency of collision is going to be less. There's just fewer in a given space, so they're going to bump into each other less often. They're still going to bump into each other. I'm still going to have diffusion or movement to the opposite side, and that's going to continue until we hit that third image at equilibrium. Notice something really important here. Even at equilibrium, I'm going to have an exchange of molecules across that membrane. They're not just going to go, oh, we're equal. Everybody stop moving. It doesn't work like that. They're still moving. It's just what we get is we get a equal rate or relatively equal rate across that membrane one side to the other. You'll see the same thing in the bottom set. So in the bottom set of images, the yellow molecules are on the left, the purple molecules are on the right, and what they're going to do is they're going to randomly bump into and off one another until we reach an equilibrium. Okay? You absolutely want to understand that, because you need to be able to talk about the rate of diffusion. And the rate of diffusion is just what it sounds. How quickly does this happen? How quickly do I diffuse? How quickly do I cross the membrane and approach equilibrium? In general, and there are lots of things that happen, but in general, I've got 3 things that I want you to know about what causes diffusion to happen quickly or have a high rate. One of those is if the diffusion distance is short. If it doesn't have very far to travel, it can reach equilibrium very quickly. It's like running from, I don't know, running from your front door to the corner of your street, or running from your front door to the corner of 2 blocks away. The shorter the distance, the quicker it happens. Okay. Diffusion area across, the diffusion area that they have to move across is large, meaning the membrane is big. There's a lot of opportunity to cross the membrane. It's like if I take 40 people in a classroom and I say, okay, I'm going to blindfold you all. You have to randomly bump into each other, and you're going to manage to make yourself out through one small normal-sized door. That's a very small surface area at which you have to precisely hit to be able to get out of the room. But what if I take those same 40 people in a classroom and I take out a big chunk of the wall, about a garage door size, and I open it up and I say the exact same thing. Okay, you guys, you're blindfolded, you're randomly going to bump into each other. How much more quickly will we reach equilibrium if they're able to pass out that larger garage door? That garage door represents surface area of which to diffuse across, and it's larger, and the rate of diffusion will happen more quickly. The last option, or the last thing that I want you to consider, is the concentration rate. So the difference between having 40 people in the classroom on one side and the difference between having 5 people in that same classroom on one side. 40 people are going to bump into each other much more frequently and much more quickly to make it across that diffusion membrane than if there are only 5 randomly blindfolded bumping around into each other to make it across. Okay, so 3 things. The diffusion distance is short, the surface area across which the molecule diffuses is large, and there are large concentration differences on either side of the diffusion membrane. Those are the 3 factors I want you to focus on when we talk about higher diffusion rates. Okay, that brings me to concentration gradient, and that's something we mentioned in that last slide when we talked about concentration differences. So a difference from one side to the other gets termed a gradient. Okay, so when we talk about substances and diffusion, we talk about them diffusing down their concentration gradient or with their concentration gradient. Okay, meaning they're moving from higher concentration to lower concentration. It takes no extra energy to do this, to move from a higher concentration to a lower concentration. No work has to be done. We're moving down or with the gradient, and we call this type of diffusion, we subcategorize it, or we include it within the umbrella term of passive transport. Any transport that does not require extra energy or the use of ATP, we call passive. All passive transport happens when we move molecules from higher concentration to lower concentration, or when we move molecules down their concentration gradient, or we move molecules with their concentration gradient. All of those are passive transport, and they do not require extra energy to make them happen. There's a converse to that, and we'll talk about it later in the chapter, and that's going to be active transport. So if you want to stick a spot in your notes for active transport, you can, but we are going to come back to it, I promise. Let's finish up with diffusion and osmosis. So osmosis is specifically the diffusion of water. It's the diffusion of water across a selectively permeable membrane. Basically, it's passive transport of water across a membrane. We call it osmosis because water is special, and it gets its own name, and that's just what you got to remember. So water will diffuse across a membrane from a region of, there's a typo on your slide, you need to fix that, from higher concentration to a region of lower concentration. Your slide says the opposite. Fix your slide. Water diffuses across the membrane from a region of higher concentration to a region of lower concentration of water. Now, the reason I told you to fix your slide is because it's talking about solutes here. So water will diffuse the opposite of the solute. So if I put sugar into the water and I have more concentration of sugar, that means I have less concentration of water. We're going to talk about this a lot in class. We're going to draw it on the board, but you really need to read carefully when you look at water and osmosis. Look to see if it's asking you about the water concentration or the solute concentration. That's really, 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 really key. When we talk about water diffusion, it's the exact opposite of what I said before. So water diffuses across a membrane from a region of lower solute concentration to higher solute concentration. You can equilibrate in your mind lower solute concentration with higher water concentration, and you can equilibrate in your brain higher solute concentration with lower water concentration. Like I said, we'll talk about that in class. If you're in my online class, you need to send me an email if that's not perfectly clear, or we need to talk about it in office hours. So, osmosis. Is osmosis a form of passive transport? Why or why not? Think about that. If you don't know the answer, look in your book. If you can't find it in your book, you need to send me a message or talk to me in class. But you need to understand why or why not that is passive or not passive transport. This image right here, students hate. And I'll admit to you that I hated it for the longest time. But it's actually a really great image. It's a really great concept, and it's a really great method of demonstrating osmosis. So, and it helps to reinforce that whether you're talking about solute concentration or water concentration, okay? So in the first image on the upper left, I have a semi-permeable membrane at the base of that U, okay? It's selectively permeable or semi-permeable. You can use those interchangeably. And on the left, I have less solute. Those little green balls are sugar molecules. And the number of little green sugar balls or sugar molecules on the left side of the U are fewer than on the right side. But notice the level of water. The level of water is equal across both sides of that U. It's at an equal height, so it's at an equal volume, which means that in this current situation, I don't have the same amount of water. At first glance, you're like, oh yeah, they're equal, they're the same height. I have the same amount of water on both sides, but you actually don't. And you don't because you have this sugar. Those sugar molecules take up space, and I have fewer sugar molecules on the left. And I have more sugar molecules on the right. So what that means is I have a higher water concentration on the left, I have more water over there, and I have a lower water concentration on the right. I have less water over there because the space is being taken up by more sugar molecules. Think about that. Look at that. Now the thing about this semi-permeable membrane in this example is the sugar can't cross it. Only the water can. So since I can't move the sugar molecules to equilibrium, I can't get equal concentrations, what do I have to move? I have to move the water. And in the upper right image, you can see that's exactly what happens. What happens is the water starts to diffuse across that membrane, and the volume of water goes up on the right, and the volume of water goes down on the left. And it's all about making the concentrations the same. What's happening is that water moves from left to right as I'm diluting the concentration of the solute on the right. I'm making it so they're same concentration. The volumes are absolutely not the same. If you look at that YouTube, they're not the same. But what I'm getting is I'm getting equal concentrations. Okay. Now, whiteboard question. List 2 or 3 causes for decreased or low diffusion rates. Read that question carefully. List 2 or 3 causes for decreased or low diffusion rates. All right, I'm going to count to 5. You're going to pause, write it out, and then I'll give you the answer. Okay. How many of you read right? How many of you read correctly? How'd you do? Your slides, our previous conversation was all about causes of higher or more rapid diffusion rates, increased diffusion rates. I asked you about lower. This is something I commonly do. So I'm not asking you to regurge information to me. I'm asking you to flip the knowledge in your head. So if a short diffusion distance causes an increased diffusion rate, then a long diffusion distance causes a low or decreased diffusion rate. So all of these answers on this slide are the opposite of what you've got in your notes. They're the exact opposite of what we talked about, but I asked you the opposite question. So just, this is my reminder to you guys to read carefully. Read questions on exams carefully, underline or highlight keywords as you're going through, and that'll help you from tripping up on stuff like that. This is easily a 3-point or even a 4 or even a 6-point if I'm asking for details, a 6-point question on an exam. Or it could even just be a 2-point multiple choice. All right, so now that you're feeling semi-comfortable with the concept of diffusion and concentrations, we need to talk about water balance of cells without cell walls. And when we talk about we talk about something called tonicity. Okay, so we'll start with tonicity and we'll apply it to cells without cell walls, and then we're going to apply it to cells with cell walls. So let's start here with just a definition. Tonicity is the ability of a solution to cause a cell to gain or lose water. Okay, that's really bulky, but that's what's in your book. Okay, so we're gonna make this easier. We're gonna throw out some of that double stuff here. Tonicity is what drives water into or out of the cell based on the concentration of solute. It's that YouTube example that we just did, but we're gonna stick a cell in there. Okay, so if I place a cell into what's called an isotonic solution, okay, in this case, the solute concentration will be the same inside and outside of the cell. Okay, the amount of glucoses per volume are going to be the same inside and outside of the cell. The end result is no net water movement across that plasma membrane. Please notice that I did not say no movement. There is still movement of water. Remember what we said about equilibrium. It still has movement of molecules. So we're still going to move water molecules into and out of that cell. We're just going to do it at an equal rate. We're not going to have an increase or decrease on either side of that plasma membrane. Okay, 2 more terms. Hypertonic and hypotonic. Okay, a hypertonic solution. If I drop a cell into a hypertonic solution, the liquid outside the cell, has a greater solute concentration. It's the side with the more glucoses. Okay, so there's more glucoses out there than there are outside the cell than there are inside the cell. What that means is I have a greater water concentration inside the cell, and what's going to happen is because I want to dilute out that glucose outside, I'm going to have water leave the cell. The cell will lose the water. So if I put a cell into a hypertonic solution, the cell is going to lose water. Conversely, hypotonic solution. In a hypotonic solution, the solute concentration is less outside the cell than inside the cell. So I have more of those glucose molecules inside the cell. That means I have a lower water concentration inside the cell, and what's going to happen is water is going to flood into that cell. All right, let's look at that visually. Okay, so let's do them in the order that we did them. So isotonic is in the middle. Top row are animal cells. So in an isotonic solution, what's going to happen is water is going to move into and out of that cell at equal rates, and the volume of that cell is going to stay relatively the same. It'll flux minutely, but it's not going to change with any significance. Move to the right for hypertonic, because this is the order we talked about them in. In a hypertonic solution, I'm going to lose water from that cell. That cell is going to shrivel up as water leaves it. Basically, I'm dehydrating the cell. I'm pulling the water out of the cell, and it's going to shrivel up. That image there is of a red blood cell, and the specific term for when that happens to a red blood cell is crenation. If you know that, that's a great bonus question for me, but you've got to understand the concept that it shrivels. All the way on the left is hypotonic, okay? And in a hypotonic solution, the water is going to flood into the cell. And I want you to think about this like a water balloon. Okay, so water is flooding in, flooding in, flooding in. The cell is expanding as far as it can, and it's going to hit a point where that cell membrane can't stretch anymore. The same way that a water balloon can't stretch anymore. And what happens to the water balloon? It bursts, right? Okay, same thing is going to happen to that cell. But in a cell, we don't call it bursting, we call it lysing or lysis. You've seen lysis before. You saw it as part of the term hydrolysis or hydrolysis, right? And that was when we used water to break apart a complex molecule into two simpler molecules. In this case, the water flooding in is going to lyse the cell, and it's going to cause the cell to rupture. Now, it's slightly different for us when we talk about cells with cell walls, and that's down along the bottom. Okay, so down along the bottom, it's different for plant cells because they have a cell wall. So in that image down there, we're going normal for a plant cell is to be in a hypotonic solution, to have water constantly moving into that cell, inside that cell wall. What it does is it keeps that plasma membrane maximized out and pushing against the cell wall, sorry about that, which helps to maintain rigidity. If I stick that plant cell in an isotonic solution, it's going to become flaccid. Okay, it's going to get a little bit limp. It's going to get softer. It's going to be that part of, I know what it is. It's when you go on vacation and you don't water your plants and you're gone for, 3 or 4 days, long weekend, and you come home and they're a little bit droopy. Okay, those cells have lost some water. The plasma membrane isn't pushing hard against the cell wall, and they're just a little bit wimpy. You water those plants when you get home, you go, oh, crud. And you water those plants, and within a day or so, they're back up to being nice, tall, rigid stems. That's flaccid. So normal in plant cells is turgid or turgid. That's in a hypotonic solution. Flaccid is just a little bit limp where we're not getting full pressure against that cell wall. That's in an isotonic solution. And then in a hypertonic solution, what actually happens is we're going to draw water out of that cell. The plasma membrane is going to pull away from the cell wall. The cell wall still exists, but we're going to pull that plasma membrane away from it so far that the cell wall can't maintain the plant. It can't maintain the uprightness of the plant, and it's literally going to crumble and fall over. That process is called plasmolysis. And that's when the plasma membrane pulls away from the cell wall. I always ask about tonicity, you guys. This is a big concept, whether you're talking about plant cells or animal cells, and I always ask it. You need to know that animal cells are normal in isotonic solution. You need to know what happens to them in a hypertonic or hypotonic solution. You need to know that plants are normal under a hypotonic situation, that's called turgid or turgid. You need to know what happens under an isotonic and a hypertonic solution. All right, let's cruise through osmoregulation. Osmoregulation happens, water, regulation, control. So this is going to happen for organisms who live in a watery environment, and especially if those watery environments are either hyper or hypotonic for that organism. Okay, you met one of these organisms when we watched the video on the amoeba and the paramecium. Okay, so those little paramecia, the two little paramecium that got eaten by the amoeba, those guys live in ponds. And that pond water is typically hypertonic to the paramecium. So here's how this works. Osmoregulation is the control of water, right? It's the control of the water balance. So it's how those single-celled organisms like the paramecium keep the correct amount of water inside their single-celled little bodies. So when we stick a protist like a paramecium into a hypertonic solution, into a pond water environment, what happens? It's hypertonic. What's going to happen? Okay, I've got water moving, yes? Here's your image. So what's happening is as water comes into this paramecium, it gets filled into a very specialized organelle called a contractile vacuole. Okay, and it fills and it fills and it fills, and that contractile vacuole will expand within the paramecium. When it gets full enough, what happens is that vacuole contracts and expels that water out of the cell. This is a 24/7, 365 process for this paramecium, because if this fails, what's going to happen is water is going to flood into this cell, into this paramecium, and it's going to rupture. Okay, so it needs that specialized contractile vacuole to maintain its life in that hypertonic environment. Here are our words when we talk about water balance for plant cells. So there's your turgid, your flaccid, your plasmolysis. There they are in writing. I'm not going to run through them again. They're right there. Okay, so this is my pause point. We'll do one last video for the last set of slides here when we pick up facilitated diffusion and active transport.